Hello everyone. Today we're going to talk about site-directed mutagenesis. First, what is site-directed mutagenesis? Site-directed mutagenesis means to cause intentional mutation at predetermined location of a gene. There are several approaches to site-directed mutagenesis. They are oligonucleotide-directed mutagenesis, cassette mutagenesis, and PCR-amplified oligonucleotide-directed mutagenesis. First, we will talk about oligonucleotide-directed mutagenesis. It is a process in which a synthetic oligonucleotide with a desired sequence change at one position is hybridized to a single-stranded copy of the gene to be altered. Here you can see a single-stranded copy of recombinant plasmid DNA, and this is the oligonucleotide that we talked about. Then there is a single change in the oligonucleotide fragment that is marked in red, that is C here. And this acts as a primer for the synthesis of a duplex DNA with one mismatch. This oligonucleotide fragment that we talked about works as a primer that is hybridized to this single stranded recombinant plasmid DNA. And when this acts as a primer to create that duplex DNA, there is a mismatch here because there was a change in nucleotide that we caused here. As a result, there is a C beside A, whereas there was supposed to be a T beside A. So this is a mismatch. So the cell wants to repair this mismatch. For this, the cell will separate this duplex DNA and turn them into two single-stranded DNA. Then the cell will synthesize the complementary strand to both of these plasmid DNA. As a result, the 50% of the mismatches will reflect the desired sequence change. Here you can see now there is a G beside C that we wanted from the beginning. This is called the oligonucleotide directed mutagenesis. So there is a point mutation caused here, as we have changed a sequence one at only one position here. Now let's talk about cassette mutagenesis. As the name suggests, this approach resembles cassette player. Like we insert a cassette inside a cassette player to get the song of our desire, similarly in this process, we insert a completely new synthetic oligonucleotide fragment, which is also called cassette, is into the DNA plasmid to get the intended gene to produce the target protein. Finally, the PCR amplified oligonucleotide directed mutagenesis. In this process, we can either insert or delete a gene of interest using PCR. Deletion are engineered by designing standard non-mutagenic forward and reverse primer that flank the region to be deleted. So we place the forward primer here and the reverse primer here. As a result, these this part of the DNA this part of the gene that we want to delete is flanked by the both primers. As a result, when these will go into the PCR, we will have a final product that will not have this flanked region because the forward primer and reverse primer is flanking this region to be deleted. For insertions less than or equal to 6 nucleotides, we incorporate the insert to the 5' end of the forward primer. For bigger insertions, half of the insert is incorporated into the 5' end of the both primers. As a result, when this goes through PCR, the overhangs will be inserted into the final product. Thus, we will have these overhangs inserted into the final gene product. So that's how you do site-directed mutagenesis. Thank you everyone for joining today's video.